I am going to invite Dr. Karsten Suhre, who's going to talk metabolomics, and for me it's a new subject.
I will try to be on time. Just let me check this one here. So I will speak today about genetically influenced metabotypes and their role in metabolic individuality. So let me just get straight into the topic. I think it's very much related, not to disease now, but what is underlying the disease, like what we heard this morning about penetrance of metabolic disorders and stuff like this. So what I want to tell you more is about what could be the underlying genetic variants, the natural variants in the population that could modify genetic diseases. So what makes us different? I think we're pretty much in agreement that what makes us different is a combination of what is in our genes, and then of course what is our lifestyle, which I show here as nutrition input, but of course that accounts for everything like lifestyle, like whether you do sports, whether you eat and drink, and so on. Getting into metabolomics, so you said you were new to metabolomics. Metabolomics basically means, what I show here is the metabolic network of the human body. So every dot is basically a small molecule that is transformed in your body, and every line corresponds to an enzyme. So this is the KEG pathway map. So what happens in your body metabolically can be displayed by this kind of a picture. And then when we talk about what makes us different, actually it's difference in the genes, so mutation in your DNA, resulting in a changed metabolic phenotype, which I show here by the red dot here. So how can we go about to figure out how are we metabolically different? The techniques are there. Whole genome genotyping, genome-wide association study have been on like, I think, 2005. And metabolomics very recently is basically the complement to genetics, scaling up biochemistry to whole metabolome characterization of what's in your blood in small molecules. So what we are interested in now is identifying how do genetic variants and metabolic phenotypes associate. And actually this is a very old topic, so you probably know Archibald Garrett. He introduced the idea of inborn errors of metabolism. PKU is a prime example of that one. But very interestingly, if you look into his book, even from 1902 and 1909, he already said that inborn errors of metabolism actually only merely extreme examples of phenotypes which are presently everywhere in the normal population. And he suggested really 100 years ago a link between chemical individuality, which today we would call rather metabolic individuality, and predisposition to disease. So if nowadays we try to link genetic variants to disease in GWAS, the problem is you go from genetic variants to very often a very complex phenotype, and most of you have probably heard about the missing heritability. So when you go into complex disorders, you find association between genotype and phenotype, but the effect sizes are typically rather small. So you're really trying to understand what leads from the genetic variant to the phenotype. And that's where metabolomics comes in. The idea is you put into the middle between the genetic variants and the phenotype a very broad characterization of your metabolome, of your biochemical processes in your organism, which can be considered a readout of merely all the metabolic processes as endpoints in your organism. So what we have done is an association between genetics, genome-wide association, not with disease, but with an intermediate phenotype, with a large set of small molecules, and from there on you can link these molecules to the disease. So in 2008, we published the first genome-wide association study between genetics and metabolomics. And just very briefly, what we really found there was what we call nowadays the genetically influenced metabotype, or part of our metabolic individuality, which is a genetic variation in a gene which is typically a metabolic active gene, an enzyme, like here FADS1 is our prototype. It's the delta 5 fatty acid desaturase. It converts fatty acids from the omega-3, omega-6 fatty acid pathway into arachidonic acid. And what we observed here is that major allele homozygotes of a specific variant in FADS1 actually have a 50% higher efficiency in converting fatty acids to arachidonic acids than the minor allele homozygotes. So this is basically very, very strong effect size for GWAS. And it can be seen as, in this case, like some kind of a valve in your metabolic capacities in your body. Or otherwise stated, if you take the population, so these variants are typically rather frequent, so with a minor allele frequency of 20%, you would expect to find about 4% myelin homozygotes in the population. And these people, given our results, could be discriminated between fast, intermediate, and slow metabolizers. So starting with this initial discovery that there would be these metabolically influenced genotypes, we continued doing GWAS with a larger panel of metabolic characterization, meaning you go into larger populations with hundreds or thousands of people, characterizing hundreds of thousands of genotypes and hundreds of small molecules in parallel. So we did a series, I can't go into all of the details, but we did different panels of metabolic screening here. Biocatis is more lipidomics based, so more lipid-oriented diseases. Then Genomics was in urine based on NMR measurements. And most recently, I will give some examples from this year. We worked on non-targeted metabolomics in a population of about 2,100 individuals. What you see here is the association study in the German CORA and then replicated in the UK Twins UK study with about 2,500 people in total. And this is the Manhattan plot that presents to you all the genome-wide significant loci where you find a genetic association between a genotype and a metabolic phenotype, a metabotype, right? So now the question is, what does this mean? So is this just an association between some genetic variants and some non-lethal changes in metabolism, or can we learn something from this? The first really interesting thing that struck me very strongly here is you find a match, in this case in 30 out of the 37 cases, between the function of the gene, where the genetic variant is located in, and the metabolic phenotype. And that's something I wouldn't expect from the start, because we do metabolomics in a drop of blood, we do genetics in a drop of blood, and they are totally different techniques. But what turns out is that the mutation is in 30 out of the 37 cases directly related to the metabolic phenotype. Like, for instance, you see here, third line proline dehydrogenase associates with proline, glutaminase with glutamine, oxoproline with oxoprolinase, and so on. And also for the fatty acid, delta 5 and delta 6, you see it's even the ratio between the product and the educt that goes with the phenotype. So there's a very close relationship between the genetic variant, the function of the gene, and the metabolic phenotype. And that can be used for a number of things. One thing is here again our FADS1 prototype. So you would expect, for instance, an interaction between different capacities to metabolize arachidonic acid and related phenotypes. And that has been shown in 2010 in a study where they showed there is an interaction between intake of omega-3, omega-6 fatty acids, the FADS1 genotype, and the outcome cardiovascular related HDL. So an interaction, depending on whether you have the one or the other genotype, gives you a different risk for getting higher or lower HDL, depending on your nutritional input. And that could have been predicted from our study before. Another already known effect is a variant in SLC01B1. It's an interaction between statins and statin-induced myopathies. And what we find here is basically that the same phenotype that gives the risk for statin-induced myopathies associates with a very specific change in fatty acids that can now be used to understand why you see these myopathies. Functional genomics. So I said there's a very clear link between the genetic function and the metabolite, and that means if you have a gene with an unknown function and a SNP, a genetic variant, associates with that gene, you could infer the function of the gene, or vice versa. If you know the function of the gene and you don't know the metabolite, you can infer the identity of the metabolite. And that's what we did here. So for this transporter, SLC16A9, it was only very globally characterized as a transporter. Its association with carnitine suggested that we should try to express this in oocytes or frog oocytes and to see whether the transporter would actually transport carnitine. And to make it sure, just to show you here, is actually we could show, based on GWAS data, that this transporter is a carnitine efflux transporter. All right, and now we come to the most interesting part, of course, of these applications is to human disease. So the question now is, all these 37 different genetic variants, they all change our metabolic composition, our metabolic makeup in our genes. So within this room, we could characterize every one of you based on these 37 genetic variants, and everyone would have his own metabolic individuality or capacity based on these 37 genes. And given that these genes all have a very strong effect on your metabolism, it's very strongly to be expected that depending on what combination you have, there would be different predisposition to complex disorders, and that's what you see. So what we have here is an overlay of these 37 loci with complex disorders from the GWAS catalog, and the yellow ones are overlaid with risk factors. So there's a very strong enrichment here for genes where you have a metabolic phenotype and also a disease phenotype at the same locus. And then there are also some other more harmless things like body hate. Having said this, the question is, of course, how do we get from the genetic variant to the complex disease? So the theory was we go from the gene to the endpoint via intermediate phenotypes. So what we have here now are 37 candidate genes where in a targeted way we could say there could be interaction between gene or gene environment. And very concretely, we have some examples here, like there are 5 different genes with genetic variants that interact with different genetic metabolic phenotypes, plus interaction with, in this case, omega-6 fatty acid intake. And the whole interacting network, in the end, associates with coronary artery disease, knowing that every single genotype has only a very, very small effect size onto the endpoint. All right, and that brings me already a bit to the end. Now, GWAS have been criticized by some people in the past, like saying, what does this bring you? We have a lot of genetic variants. They are very small in effect size, so what do you do with this? I think I've shown you already before. You can do functional genetics. You can look for gene-gene interactions. You can try to explain why complex disorders act the way they do. Why don't you have full penetrance in other disorders? But especially an idea is also from GWAS to say whatever gene associates with a phenotype, it's a target for a drug. And in the case of metabolomics, you could now think about using it for what you would call rational metabolic engineering, meaning that every of these 36 variants, and in the meantime we have some more from new studies, is actually a target where you could intervene in the sense of saying, I put on a valve on these different pathways. So imagine this here is your metabolic pathway. Everything is changed in your body. You have the inputs there to the left, different things getting in and out of the body, ending up in different diseases. Now, if I want to change the metabolic makeup of a patient, I don't have a drug that can just kick out arachidonic acid or just reduce glutamine or whatever. But given the knowledge we have here now, we can design strategies based on the different genes that associate with the metabolites. We know the valves and can now target, maybe in a combined way, different enzymes and transporters, so that in the end we could engineer the metabolism of a patient in a way we want to do that. All right, that brings me to the end. So I hope I have convinced you a little bit that metabolomics is a phenotype which is very much disease-related. Surprisingly, strong matches between metabotype and gene function show us that there's a very strong functional approach to this idea. We have surprisingly large effect sizes, frequent genotypes. So that means if we look for combination of genotypes here, it's not rare variants where maybe 1 in 1,000 is concerned. Here it's 5 in 100. So it's really not a disease. It's more like the background that defines a penetrance. We have a strong overlap with GWAS loci, and the numbers are actually increasing with new GWAS running. It's an intermediate phenotype to understand disease. Gene and gene environment interaction are statistically very hard to tackle at the population level, but if you target them, you don't have to combine millions of genetic variants, but maybe only 50 or 100, then it may become treatable to look for gene-gene interactions. Functional genomics, of course, understand how do genes that we don't know yet really function, and then rational metabolic engineering. All right, so that brings me basically to the end. Something I could not talk about today, but which is, of course, the next step also to consider is how do you get from genetic variant to the phenotype? So there could be different regulatory steps, and one we recently studied and published is epigenetics. So we now also started doing epigenetic association study with metabolomics. We can then see how do genome methylations change your metabolic phenotype. All right, so I hope I stayed somewhat in time. I should acknowledge many of my colleagues, especially from the German and the English studies, especially my colleague, of course, Christian Giger, with whom I did this the last 5 years. And with this, thank you very much for your attention.